All right, so what I want to be talking about is development of tools for multi-photon imaging of mouse spinal cord neurons during locomotion. And it's going to be a bit of an update and some of the future plans that you plan for this project here. So I started off in electrophysiology, studying small circuits, and wanted to understand spinal cord injury. And then needed to, when I went over to Ron Harris-Warwick's lab, we did a lot of spinal cord, slice of electrophysiology. But in order to understand the ramifications of spinal cord injury, you need the physical ramifications rather than just the intrinsic changes in neurons. You'll need to understand what goes on in an entire animal. So that's why I ended up joining Schaefer Nishimura lab, because they're developing tools to understand deep imaging in brain and spinal cord tissue, as well as other tissues in the mouse. So what particularly drew me towards the lab was the use of 3-photon excited microscopy. And that's what I will be using to have a deeper look at the neurons in the mouse spinal cord. In addition to that, I'll also be talking about some of the adaptive optics, as well as the meat of the project, which is the automatic kinematics and gait detection. So, right. So I'm a neuroscientist mainly, not really a physicist or optics person, but I've definitely gotten into optics since I've been in Schaefer Nishimura lab. But the importance of locomotion really is for survival of the animal. And survival of the animal, they'll need to rely on circuits in the brain as well as the spinal cord, but mainly the spinal cord has circuits called central pattern generating neurons in order to exhibit a variety of these types of behaviors that we see here on the screen. So central pattern generating neurons, they can mainly be divided into two different types, cell autonomous and network-based. Those that use network-based bursting mechanisms. So what that basically means is that, for example, cell autonomous networks, these have a specific group or a single cell that has intrinsic ion channels and neuromodulator receptor combinations that allow it to burst on its own. Network-based mechanisms are circuits where two or more neurons work together in order to generate bursting type output. So for example, you can imagine when you're walking, you're going to need circuits, spinal cord CPG circuits, in order to move your left and right leg, to run up and down stairs, to walk, etc. So this is a brief overview. Most of the initiation of essentially generating circuits come from electrical and motory circuits in the midbrain. So for example, if you stimulate the mesencephalic locomotor region in the medulla, in the midbrain, sorry, synapse onto circuits that produce serotonin, mainly serotonin, in order to trigger spinal cord circuits in the spinal cord CPG circuits in the spinal cord in order to generate locomotion. So this just summarizes a lot of the older sharp electric physiology work. So what we have here is patch electrode electrophysiology combined with suction electrophysiology in order to answer the question, what are the outputs of these type of circuits in a fictive locomotor setup? So in order to trigger fictive locomotion, you need a cocktail of neurotransmitters, chemicals, etc., like serotonin, norepinephrine, NMDA, etc., in order to make spinal cord section generate this fictive locomotor output. So as you can see here in the L2 and L5 motor routes, you can see that there's there's electrophysiological locomotor output, fictive locomotor output. And when you patch onto cell well, this is the dorsal section of the spinal cord that's cut off. And when you patch onto one of these neurons, you see that they burst in time with the fictive locomotor output of the ventral motor root. So this suggests that there are spinal cord neurons, that interneurons that are active during fictive locomotor output. This just gives a brief, I talked about this before, but this just gives a fairly brief overview of what, how these neurons, spinal cord interneurons, are divided. So they're divided mainly by transcription factor, different transcription factors. For example, V2A are segmented by CHOX10, V1, and GRAIL1, etc. And they're also yeah, that's basically it. I don't want to go too much into that. So transcription factors are the main way that these neurons are divided up. And this is a cartoon of what spinal cord basically looks like. We have the dorsal section, which contain mostly the sensory neurons, which can be easily viewed by 2-photon excited microscopy. As you go deeper, you get to where the interneurons are. And these are the main neurons that are involved in patterning the locomotor output. And they're, as I said, characterized by the different transcription factors, V2A for CHOX10, V1 and GRAIL for V1, etc. So all the motor neurons, interneurons, and sensory neurons work together in order to generate central pattern generator locomotor output. And this can be done without sensory feedback, as I was showing in the fictive locomotor output study. All right, so let's get to some of the microscopy stuff. So in order to even look at these neurons, we'll need to develop an optical window for the optical access to the spinal cord. This was developed by Farrar et al. Back in 2012. So this is the first first version of the window for anesthetized imaging. It has the little wings on the side in order for the little bars to hold on to the window itself. So as you can see here, this red line, that's the dorsal spinal vein. And on the either side of the cord here, that's where we can see some of the white matter and the gray matter. And it's a lot easier to look through the gray matter. The white matter becomes extremely problematic, especially with 2-photon, because the myelin scatters light quite a bit. But I'll get to I'll get to a way through which we can get around the aberration that's caused by the myelin. So in order to, as I said, implant the window, we do a dorsal laminectomy, which permits chronic imaging. This is just a schematic of what the spinal cord imaging window looks like. So on either side of it's basically a spinal cord fusion, where we have on either side of the spinal cord, we put 2 bars to stabilize the spinal cord and screw down the top plate in order to implant the glass window. Well, all right, let me be a bit more detailed. So we remove the top part of the vertebral bone for the dorsal laminectomy, and then put an optically clear, quicksil, and then put the glass over that. And this shows an example of a mouse that's, this was one day after. I was going to give it some drugs, some pain medication. And as you can see, once the laminectomy is done pretty well, the mouse can ambulate normally. I mean, it's not going to run around fast without pain, but you can see that as it's able to go around the cage as normal. All right, so in order to view, all right, so once the spinal cord window is on, we have to use lasers in order to excite fluorophores in the spinal cord in order to see the structures. There are also, and since it's a 3-photon microscopy, there are also endogenous labels like TSG and SHG that enable us to see different structures. TSG mainly helps us to look to see what the myelin looks like. So this here is an example from a review paper by Yuting Cheng, who's already graduated from the lab, comparing both 2-photon excitation, 3-photon excitation. It's pretty much evident to see why 3-photon excitation is required to see the deeper CPG neurons, mainly because those neurons lie beyond the 300 to 400 micron threshold, depending on where along the spinal cord you are. So the benefits of 3-photon excitation for what we're doing is essentially to have additional clarity, resolution, as well as depth. All right. So this I showed before. Adaptive optics mainly requires a deformable mirror. I'm going to get to that. And so, right. And because of the curvature of the mouse's spinal, so this was built by Mike Lamont. So because of the curvature of the mouse spinal cord, we have a rotating objective, and that enables us to make the mouse to image while the mouse is able to run comfortably. Because some of the initial trials that we had, because the objective was perpendicular to the imaging window, the mouse could have ambulated, but it was the ambulation and locomotion was not quite natural. So, and as a neuroscientist, the imaging, the GCamp activity that we see from these neurons, we want to make sure that they come from natural locomotor behaviors, because if it's just random GCamp activity with errant looking behavior, you can see that, there's activity in these cells during locomotion, but it's not really going to be natural, and it's not going to tell you exactly what these spinal cord CPG neurons do during a real locomotor behavior. All right, so back to my setup. So so they're hot swappable optical filter and dichroic tubes, and the final iris is used to reduce the average power delivery over the sample. So the deformable mirror, I think most people know what a deformable mirror is. Basically, the little actuators behind silver, polished silver sheet, and these can bend the mirror to the desired configuration in order to compensate for different wavefronts and compensate for different wavefronts. Okay, so Mike also developed a deformable mirror GUI. So what that GUI does enable us to tune the different Janicki polynomials in order to adjust the mirror position as well as shape in order to help us increase the resolution and boost signal as we go deeper into the spinal cord. All right, so on the scanning iris, this is pretty important. And so in our initial imaging trials, awake imaging trials, what happened after longer scanning sessions, get burning. We observe burning on the edge of this, on in the imaging field. This is happening because as the laser scans across the field, this turnaround here, the lasers not essentially parked, but there's more time for it to stay within a certain region. So along the edges here is where you'll get burning after long imaging sessions. So Mike ended up adding this final iris in order to help us mitigate that burning. So this is some of the, I should probably skip this because this is some of the older data to show that with more dorsally located neurons, we're able to observe calcium transients. These two aren't time locked yet. These two aren't, these two aren't time locked. This was an older version of the treadmill. This was a flat version where we had the imaging window perpendicular to the objective. Yeah, the mouse is moving around. It's not really ambulating a lot, but we're able to observe during this time point that we can see that there are calcium transients, some of these dorsally located CAM-K2, GCAM6S neurons. All right, so we had to make some adjustments to stabilize the window because you can imagine that the locomotor circuits for locomotion located in the L4 to L6 region of the mouse spinal cord, which control the hind legs. So we literally have to put the window close to the over the hind legs. So I don't know if you guys looked at mice running or squirrels running outside, that part of the mouse moves a lot. So the potential problem there is that is going to occur. It's motion artifact, and especially in the Z direction, it's pretty much a nightmare. So we can also correct for motion artifact in the XY direction, which thank goodness that can be done. So I ended up, we ended up designing a new version of the top plate. This top plate, along with different C-clamps, so a lot tighter tolerances, allows us to stabilize the window a lot easier compared to this earlier version. So yeah, so this was, right, so this is an older version of the linear treadmill. The older version of the linear treadmill, there was some pros and cons to that. The pros is that for this version of the treadmill, there's a lot more data that we could extract from the treadmill. There are built-in sensors in this treadmill that allow us to extract data such as distance, speed, etc. And we made some additional modifications to modify the speed of the treadmill as the mouse ambulates. So this here is actually forced running, which required a lot of training for the mouse. And that additional training, so after the window is implanted, takes about 5 to 7 days for the window to heal, and then an additional 3-4 days of rigorous spine fix and free running and tail fix training. And during that time, there's a chance of bleeding that will occur. And once bleeding occurs, you aren't able to see anywhere in the window, and there goes your time. So for this version, version, there are some definite advantages, but the time we needed to find a way to make the mice be trained a lot faster in order to get the data and to get the data.
Yes, it was.
Yeah. So
when I was seeing, when I'm looking at your movie, also came to mind, what I was experiencing in my laboratory, and we were struggling a lot to get the mouse, and it was never a natural locomotion. So we did basically, the spinning wheels that they use as a toy for the,
Yep.
basically,
Yeah.
after using that one,
Mm-hmm.
Basically housing them with this, then, they were running a little bit better. But still, to us, it was a source of big headaches. Sorry for the observation in the middle of your talk. Just wanted to share the frustration.
No, that's perfectly fine, and I'm glad that you mentioned it, because I'm going to get to that later on. So this training paradigm, we tried it without the home cage flying saucer treadmill for a while, and then we ended up putting that flying saucer treadmill in the cage in order to facilitate training, just to get them used to running on an unstable surface. Let's see. All right, so in order to quantify, this isn't exactly optics. All right, so let's talk about this. So we need a deep lab cut in order to measure the limb kinematics, because the overall goal of this project, it's not just developing new optics, but we need to develop new optics in order to answer the question, what's the activity of the CPG neurons that are involved in spinal cord locomotion in vivo? There's a lot of older data, as I showed with electrophysiology, but it's in slice. Some of it's in actually neonatal spinal cord preparations, but it's not. It doesn't give you a full picture unless you have the entire mouse. So we use the deep lab cut in order to track different points on the limb, in order to correlate the limb kinematics with the calcium imaging data that we're going to get. So how do we do that correlation? This is some of the work that Samantha Kraus, she's actually a grad student here now. Samantha Kraus got, which was the hip, knee, and ankle angles. All this is just raw data that we can use to put into Matt Isaacson's. He's a postdoc who's helping with the coding pipeline. So we can put all that data along, all that kinematic data with the calcium imaging data in order to line that all up to determine the function of these particular neurons during the gait cycle. All right. So, yeah, all right. So most of this, so the new stuff, actually, so because of the quote unquote frustrations of working with the linear treadmill, we ended up, I had to try something where it's less sophisticated than the linear treadmill with all the electronic stuff added to it. I just had to use the same treadmill and just put together, the same posts, graduated posts, along with the spinal cord imaging clamps. This is just a prototype version on the side here. And after training, the mice are able to, this is a good example. One of the good examples where a mouse is able to run voluntarily on the circular treadmill. So in a sense, we kind of had to not really start over, but start over the kinematics, the deep lab cut kinematics section of it. So I had to acquire more of the training data in order to train a new network, in order to correlate that to the spinal cord imaging. But in addition to that, and I'll get to something else optics related soon, because there are some additional complications to using this angled treadmill versus the flat treadmill. Oh yeah, that's just a different angle. All right. So along with Maria Guzman, she's one of the other REU students who helped with the new version of the treadmill. So we had to rig up a spine fixed, a way to spine fix the mouse on this angled treadmill. Again, we had to determine what was the major differences between the spine fix and the tail fix running paradigm. So this is an example of the mouse underneath. This is underneath the microscope. And these are the dots that we used from the deep lab cut. So we trained and filmed these mice underneath the microscope, so we can have an idea of how they behave during imaging day. This is voluntary running. So the new training paradigm was that we had to do the same implanting the treadmill, wait 5 to 7 days for the mouse to heal up, and once the mouse was back in the treadmill, we put the flying saucer treadmill into the home cage, and they're trained on the flying saucer treadmill underneath the microscope for 3 to 4 days. So there are going to be some obvious differences when a mouse is spine fixed versus free running, and I'll show some quantification of that afterwards. The mice are able to run pretty well while, I mean, they love doing this. So but as you can see, there's a difference between having a specific section of the spine fixed for imaging versus having it free run on the treadmill. So this is some of the data that Maria got. So what she quantified here was the gait frequency histogram comparing the free running and spine fixed mice. As you can see, there are some differences in the overall frequency of running between the spine fix and a free running mice. I mean, this just tells us that because due to the nature of being spine fixed on the treadmill, there's going to be some slight differences in the way the mice run. So that's where most of the some of the work has been put into just getting this new system up and running with the less sophisticated treadmill. But I think in the end, having the mice run comfortably is the best thing because when we initially tried this preparation, sometimes the mice would just sit there and not do anything, and we'll be running the laser over the imaging spot and it will just burn after a while. So we needed a way to have the mice first run over a motorized treadmill, and now where we're at, having the mice run voluntarily. And actually, the good thing about the flying saucer treadmill is that because of the way it's angled, gravity sort of pulls the mouse down, so it triggers the spinal cord circuits for the mice to voluntarily run. So they just constantly run and run and run until they get tired. And in addition to that, we tried angling the leaner treadmill. That helped a bit, but the training paradigm was just what was holding us back. So this shows now what the new wireframe is going to look like for when we put together all the data. All right, I should have shown this earlier. So this is just the new, all right, so this is, yeah, so after Maria left, actually no, this was one of the frustrations that we had. So in order to understand what the spinal, so we can only, so the imaging field is only so big that we can only image only one side of the cord. In the spinal cord, there are two sides. One controls the left, one controls the right, and they're inhibitory neurons in between the left and right between the left and right side of the spinal cord in order to facilitate left-right alternation. Because in order for different sorts of behaviors, for like walking, you're going to have to inhibit the one side while one side is active, and then that cycle just keeps on going on. For example, if you're hopping, you'll have both sides synchronously going. What does that all mean? So if we image only one side of the spinal cord, we're not going to get no. So we can image one side of the spinal cord, yes, but in order to get a full picture of what the overall kinematics, we need to image both left and right sides of the mouse. So due to the nature of the flying saucer disc, it just made it, and the detection optics box here, it just made it a bit more difficult to position this camera on the side here. So we weren't getting a full picture of what the left side was doing. So along with Rick Zirkel, another graduate student in the lab, we had to put together a new flat version of the treadmill. So this just pops off that older version of the flying saucer in order to help us image both left and right sides of the mouse. Right. Okay. Yeah. So yeah, this project is mostly getting the mouse to behave and adapting the setup so that the optics can work well. So when I say the optics have to work well, we have the tilting objective that I mentioned before. So I didn't have the objective mount in here, but because of the nature of this treadmill here, the objective mount will have to be tilted in order to image the spinal cord. So this is some of the preliminary imaging that I did with the tilting objective with an awake mouse. All this is just really here to show us that the imaging plane is just going to be tilted, angled the same way that the objective holder is going to be angled. All this really, it's not going to be a big deal, but all this really does is going to alter the way we navigate the cord, because normally we'll have the dorsal spinal vein going from left to right here because it's angled, and the way the optics, the mirrors are placed. Mike knows all the answers for that. It's going to rotate the imaging frame slightly. And in addition to that, the tilting of the objective and the imaging window, it's gonna, it's no big deal. It's just gonna make the navigating just taking stacks and navigating the cord a bit more challenging, because the objective not only has to go in one direction, has to go in two directions, basically in the Z and the Y direction. So yeah, so overall, we were close to the point where it's time to collect the data from the mice that we have. We have the Chalkston mice, and we're doing viral injections of GCamp into two other lines, SIM1 Cree and the EN1 Cree mice. These have different CPG neurons labeled in the spinal cord. So I think we've pretty much gotten through all the little kinks that are gonna, gotten through all the kinks that are gonna impede the way the mice runs, as well as mice run, as well as the imaging difficulties. So 3-photon, we image using we started at 1300, but we were imaging 1320 this time because of Chris Shu's paper. The adaptive optics, that's pretty much working now, so it's no problem. Most of the work has been being put into the automated kinematics and gait detection for ambulating mouse on the new spinning disc treadmill. So that's, so I'd like to thank Chris Schaefer and Nozomi Nishimura for all the help on this project. Matt Isaacson, a postdoc who's helping to put together the code for, he's putting together the code for the optics, the GCamp, and the GCamp imaging, as well as the kinematics that we get. And Mike Lamont for building the scope. Rick Zerkel is the grad student who's helping me on this project with, some of the coding for the speed sensor, as well as helping to build the new treadmill. Yuting Cheng, he put in quite a bit of work a lot of the work at the start of this project to help get it going. He's already graduated from the lab. And Daniel also helps with the coding, and 3 RU students, Maria, Samantha, and Kelsey, for their great efforts in helping to get this project going.